Hi, my name is Joe Layton. I'm the Director of R&D Bakery at Puratos, and I'll be walking you through his technical training that covers different aspects of non-GMO and organic rules and regulations. I'd like to start with an overview of what we typically see in the industry, ranging from cleaner label, which is the least restrictive, to organic, which is the most restrictive. When it comes to regulations, there's really no regulation behind cleaner label. Each manufacturer and each retailer has their own guidelines. We typically see Whole Foods being the standard in the market, but more and more each company has their own guidelines as to what clean label actually is. With non-GMO, there's now a few different regulations and also rules in place. We have the new USDA bioengineered foods disclosure, which was released in December of 2018. There's also third-party organizations like the Non-GMO Project being the biggest that certify products for non-GMO status. And those are typically done through technical administrators. The main ones for non-GMO project are Food Chain ID, NSF, SCF, SCS, and where food comes from. And finally, we have the organic, which is really the most restrictive. This is a very well-defined program. The USDA regulates organic status. Products can only be produced on a certified line. There are roughly 80 different USDA accrediting agents. Puratos has several plants that have been certified as organic. Our Kenosha, Wisconsin chocolate facility, as well as several lines in our Pennsauken, New Jersey facility have been regulated or certified for organic production. Here's the agenda for the rest of the presentation. We'll start with the GMO BE section, and then we'll go through definitions and some common terms, the impact on labeling, and then dive into GMO technology as it applies to enzymes specifically. Then move on to organic with a similar flow, and end with a few examples on organic calculations that will hopefully make the whole calculation setup more clear when you're going for an organic certification. So, starting with the non-GMO section, or the GMO section rather. GMO stands for genetically modified organism. So that means it's something that relates to organisms. Another common term is bioengineered or bioengineering. This also generally relates to organisms and is more or less interchangeable with genetically modified organisms when it comes to modification for use in at least the food industry. Another term you might hear is protein engineered, and this is actually referring to a molecule or a novel protein. When we say novel protein, we mean a protein that doesn't necessarily exist in nature. It was created in the lab. So a few examples here: a plant, animal, or microorganism that's had its DNA modified would be considered a genetically modified organism. A molecule that has been synthesized from DNA source that was modified. So the molecule as such is the same as what you would find in nature, but the DNA that was used to produce it was in some way modified. That would have also come from a GMO organism. And then one thing I want to call out is that there's no proven health risk for GE or GMO or BE ingredients. There are several places in the world that are taking a precautionary stance, like Europe has banned the use of GMO ingredients for human consumption, but they're still allowing animals, from what I understand, to consume GMO products. So why would we use GMO ingredients? Biologists modify the DNA of plants for a number of different reasons. Sometimes it's to make them resistant to different herbicides so they can grow more efficiently. To make them grow faster, so adding a gene for a growth hormone, for example, to make them bigger or get more yield. In other cases, to give special properties like reduced browning. In the bakery world, specifically related to enzymes, biologists are modifying these DNA sequences for certain reasons. So in some cases, it's to make the enzyme more active or to make it less sensitive to conditions like acidity or sugar content, or to get something that works differently, so at a different temperature, for example. And another reason is to use host organisms that produce the enzymes more efficiently. Often the organism where the enzyme is sourced from is not a very efficient producer of that particular enzyme and may or may not even be safe to culture for production of the enzyme. In agricultural products, really better performance is one of the main drivers. We mentioned that browning example where you could reduce browning by changing the enzymes that are in a particular fruit or vegetable. Here's an example of the Arctic apple variety that basically doesn't brown as quickly as a standard apple. With enzymes, you get improved performance in softness, moistness, and resilience because you can make those enzymes more efficient and work over better conditions. Sometimes you can get increased gluten development, depending on the type of enzyme that you're using, increased oat tolerance, more volume. You can identify enzymes that work at different temperatures than your standard enzymes. So for example, you could identify a xylanase that works at very low temperatures and gives you the impact in a frozen dough where your working temperatures are usually lower than what you typically find. Another example would be a protease that works only at higher temperatures, so it has no impact on dough rheology, but then impacts the baking performance, the texture of that product after baking. When it comes to the USDA standard for bioengineered foods, the National Bioengineered Food Disclosure Standard was published in December of 2018. Basically, it states that any food that is produced using bioengineered ingredients must have the declaration. I'd like to read the definition here, specifically the part that's underlined for what the USDA considers a bioengineered food. It's those that contain detectable genetic material that has been modified through certain lab techniques and cannot be created through conventional breeding or found in nature. So the bioengineering disclosure really is a two-point test. It's does that product contain detectable DNA, and has that DNA been modified? So a good example of this is soybean oil. Soybean oil in the US typically comes from GMO soy crops, but because the oil is so highly refined, there is generally no protein left in that product, and there's definitely no DNA. That ingredient would not be considered bioengineered according to the USDA bioengineering standard because the DNA is not detectable, even though the DNA had been modified at some point. So in terms of implementation, it has actually started already voluntarily, and then mandatory compliance starts January 1st, 2022. So there's still a little bit of time left, but it is coming up quick in terms of when companies are going to need to comply with this regulation. So what types of foods need to be regulated? There's a link here to the different list of foods that are going to be required to be labeled. Also, there are some exceptions. The incidental additives and ingredients in which the material is not detectable are excluded from this, as we mentioned earlier. There are a couple of different options on how on the packaging you'll be able to declare that the product contains bioengineered foods. You can use text. There's a symbol that's been created, both a full color and a black and white. There's provisions for doing electronic disclosures as well as QR codes, for example, in the packaging. You could have one QR code and then update the link where that product goes to, or where that link takes you to. And then calling out for bakery, specifically enzymes, because they do not contain genetic material, are basically exempt from the BE disclosure. Thinking about the Non-GMO project, Non-GMO project works on looking at the risk levels of different input ingredients, as they would call it. Anything from high risk, where it's known that that product is mostly commercially sourced from GMO crops, to low risk products where there's not much commercially available. And then you've got no risk ingredients like salt, for example, where there is no genetic material. It's not derived from a biological source. And then there are several ingredients that are being monitored currently where there's research being done to release modified versions of these crops, but so far they're not in mainstream use. The most recent version of the Non-GMO project that was released was version 15 in July of 2019, as of the recording of this, which is June in 2020. Enzymes are out of scope for the Non-GMO project, provided that they're not on the label of the product. So enzymes can be considered a processing aid, which means they do not need to be declared on packaging. If you choose as a manufacturer to include the enzymes on your label, then the enzyme must be non-functional in the final product. So the enzyme needs to have been denatured in the oven during the baking process or during whatever process that food is going through in order to be exempt from certification or exempt from disqualifying you from certification from non-GMO project. What this means is that newer generations of enzymes are allowed in non-GMO certified products, which in terms of performance means generally you can get a superior product. The majority of the enzymes developed in the last 20 years have been developed using GMO techniques, which has allowed us to develop xanases that give more tolerance and more volume, the lipases that give more softness and fineness, amylases that are much better and more efficient at prolonging shelf life and adding softness and moistness. So now non-GMO projects certified products can have the same type of performance technically as the standard products, which in early days, the standard was a bit of a hurdle because you were limited to enzymes that were only compliant with basically organic standards because they had been produced without GMO techniques. Diving in a little bit more into enzymes specifically, enzymes are molecules, they're not organisms. So by default, they really can't be GMOs. They're also proteins. There's no DNA in an enzyme. And during the baking process, enzymes are denatured, making them unfunctional in the bread. So that means that in most cases, at least for the enzymes that are sold by Puratos, we know that the baking curve denatures these products as long as they're baked at sea level. There can sometimes be concerns with enzymes that are baked at high altitudes because the finished bake temperature is lower than what the denaturation temperature is for the enzymes. So it is something to take into account is if you're hitting that internal crumb temperature of around 200 to 205, in almost all cases, the enzymes are denatured, but there are some unique situations where you may have an enzyme that's not fully denatured during the bake. Proteins in our bodies and animal bodies, every living organism, are responsible for a number of different functions. As we mentioned earlier, enzymes are a type of protein, and enzymes allow a wide variety of biochemical reactions to occur. A few examples: enzymes help with digestion of our food. They help with production of energy in our cells. But how are proteins made? Proteins are basically synthesized from DNA. The DNA is read, it's converted into an RNA, and then the molecules in our cells will translate that RNA into an actual protein by combining different amino acid sequences. Once those amino acid sequences are combined, they are going to form a specific shape, and that will become the protein that's in our body. Enzymes can be a little bit tricky in terms of what classifications that they fall under. So I'd like to put out basically 3 levels of technology that's used for enzymes. You can have a non-bioengineered enzyme that is produced with non-bioengineering techniques. So this is, you've identified an organism in nature that produces an enzyme, you culture that organism, and then you harvest the enzymes. The next thing you could do is you could have a non-BE enzyme that's produced with BE techniques. So you take that same DNA sequence from that original organism and you put that into a producing organism. The molecule or the protein that's produced as a result of that is identical to what would have been found in nature, but the organism is different. The DNA had been artificially put into this new producing organism. And then lastly, you can have a protein engineered enzyme that is produced with BE techniques. So taking that DNA that was found in nature and changing some of the base pairs to get a specific functionality. And now you have what we call a novel protein, one that doesn't exist in nature. It was basically created in the lab and now is being produced. So there again, when we're looking at the way proteins are made and enzymes, you start from a DNA sequence, and that's where if you modify a DNA sequence, that would be then translated when it's made into RNA. And when that RNA is read and it's producing a protein, that protein will now have the different structure because of the modification of the DNA. I'd like to give the example of a few different drawings. I would love to take credit for this artwork, but it was done by a colleague of mine, and you'll see why I'm so excited to share his amazing work with you. Here we have the example of a bacteria, and we'll call this bacteria L for lazy. This particular bacteria has one copy of the gene that produces a specific enzyme that we want, but he can only make, let's say, 4 copies of that enzyme, which isn't very efficient for production. So what we will sometimes do is we'll take that piece of DNA and we'll put that into a producing organism. In this case, we've got bacteria S for speedy. And now this is also a non-BE protein, but we're using BE techniques to move that DNA into a new organism. In this case, bacteria speedy can produce, let's say, roughly 10 copies of that enzyme. But that still really isn't fast enough for what we want to do and be able to have a commercially viable enzyme. So the next level of BE techniques is then to take that same gene and insert multiple copies of it into the DNA of that bacteria. And this way we can significantly increase the number of enzymes that are being produced and the efficiency of that enzyme or that bacteria as a enzyme producing organism. So by multiplying the number of copies, we can increase the production by, let's say, 10-fold to get to 100 different copies of enzyme. Still the original protein that was found in nature, so not a novel protein, but using BE techniques to accelerate the production of that enzyme. And then finally, we can go with protein engineering, where we take that specific DNA strand and we tweak it to get, let's say, a little bit more tolerance to heat so that that enzyme works for a little bit longer time in the bake and gives you a bread that stays softer longer because the enzyme was more efficient inside of that bread. We haven't changed the number of copies of the enzyme, but we are now producing a protein engineered enzyme with BE techniques. So I hope this illustration makes the different levels of protein engineering and genetic engineering clear for enzyme production. One thing I'd like to add is another reason outside of efficiency for producing with specific organisms is also for safety. The producing organisms that we use for taking the DNA and putting it into for production are very well studied. The genomes are mapped out entirely. And we use those organisms because there's very little risk, if any risk, of producing other chemicals or other enzymes that we don't want to or could potentially be harmful for a person to consume. So there's also a safety aspect of using genetic engineering because if you take that host, the original host organism, it may produce what we call sometimes a soup of different chemicals. Some of those might be detrimental in baking. Some of them might even be hazardous to humans. So by putting that DNA into a producing organism, we can ensure the safety of the product that's being produced, as well as produce it in a very efficient and controlled manner. All right, moving on to organic. So organic, as we mentioned in the beginning, is USDA regulated. This is really a production supply chain certification. So it goes all the way from the farmer to the producer. Products can only be produced on a certified line. Again, in Puratos, we have a few different certified lines in the US, specifically our chocolate facility in Kenosha, Wisconsin, as well as our Sapori and Sofrian lines in Pennsauken. Some of the main restrictions around having an organic certified product is we prohibit, or the regulations prohibit the use of GMOs. You can only use natural flavors, and the caveat here is only when organic versions of those natural flavors are not available. No synthetic pesticides or herbicides can be used. You cannot fertilize with sewage sludge, and no growth hormones or antibiotics can be used in the raising of those crops or animals. There are 3 main categories of organic certification: 100% organic certified, organic, which is 95% or higher, which is typically the case for bakery, and then made with organic ingredients, where you have to have a minimum of 70% organic ingredients. Now, there's a few more complications beyond just the number of organic ingredients, and we've tried to break those down in the chart here. With 100% organic ingredients, you're mostly limited to agricultural products. So single ingredient type products like a fruit or a vegetable. When we're talking about bakery products and most baked products in general, we're typically shooting for the 95% organic. You're going to have by default ingredients like salt and leavening acids that are not going to be organic. So they're going to count against you slightly, but achieving that 95% is usually the target for most baked products. Now, as we mentioned, everything here needs to be produced on a certified line and following those guidelines. The next level is made with organic ingredients. So here you must have between 70 and 95% organic ingredients. All of the non-organic ingredients need to be on the approved list, and that also goes for the organic standard. There's an approved list, and we'll get to that link a little bit later. But it's not that you can use any ingredient in that 5%. There's only an approved list of ingredients. Otherwise, in terms of requirements, the requirements between the 95% and the made with are basically the same. You still have to be produced on a certified line. You still have to follow all the same regulations. It's just that if you can't achieve 95%, this is the next best thing. And then finally, this really isn't a level of certification, but it's a way to use organic ingredients, is when you have less than 70% organic ingredients. You can call these out in your ingredient statement, but you're not allowed by the regulation to call them out on the front of packaging, for example. So the only place where you can declare the organic ingredients is in the ingredient statement, where you could say, I have organic wheat, regular butter, for example, and then organic color. You wouldn't be able to say on the front of your bag, made with organic wheat, because you haven't hit the 70% threshold. So a few examples of different organic products. The lettuce in the upper right there would be an example of a 100% organic product, because it's a single ingredient. It's just the lettuce. There are different regulations on which size font can be used and how it must appear on the actual packaging, and you can find that in the standard. When you have the 100% organic or the 95% organic, you are allowed to use the seal on the packaging, the USDA seal. And then in the bottom right, we have an example of a 95% certified organic bread. So this rosemary bread, you can see it has the seal on it, and that's how you know that it's at least 95% organic. So when we're doing the math for organic, as we mentioned, in most cases, we're shooting for 95% with breads or baked products. So 95% of the ingredients must be organic certified. The water and the salt are excluded from the math. And I have an example here we'll walk through that'll hopefully make that a little bit more clear. As I mentioned earlier, there's a list of allowed and prohibited substances for organic that other 5% can be made up of. So it's not that you can have 95% organic ingredients and then use specific emulsifiers like datum, for example, in an organic certified product, because datum is not on the allowed list. Ingredients like baking soda or citric acid, for example, are on the list, and there are quite a few different ingredients on the list, but you'll need to check that when you're actually doing the math to see if your product qualifies for the organic certification. And this is the link to find that list. So talking about examples of organic certification, here is an example of a recipe that has 50% organic flour and baker's percentages, 50% organic whole wheat flour, 70% water, 4% fresh yeast, 3% organic gluten, 2.2% salt, 4% organic honey, and then 0.5% of the Puratos S500 organic improver. So we have a total of 183 pounds. If we take everything in this recipe and we look at excluding the salt and the water, we have 111.5 pounds of ingredients we need to evaluate. If we take that 111.5 pounds and we convert that into percentages of 100, we end up with 44.8% of the two flours, 3.6% of the organic of the yeast, 2.7% of the gluten, 3.6 honey, and 0.5% of the 700 organic. that leaves us with a total of 96.4% organic ingredients. Now, one caveat here is on the fresh yeast. As of today, fresh yeast is questionable whether it's okay for use in organics or not. More and more organic yeast is available and commercially viable. So you'll have to check with whoever is certifying your products if they're going to allow you to use regular commercial yeast or if you would be required to use organic yeast. In this situation, we're assuming that we've been given approval to use conventional yeast. So we have 96.4%. The water has been excluded. The yeast, we're getting 0 credit for that because that is non-organic. So this product does qualify for organic certification because we have 96.4% and all of the ingredients are allowed to be used that aren't organic. So in the next example, very similar recipe, but we've replaced the SF100 Organic with SF100 Green Acti Plus, which is a clean label improver. So that only contains enzymes. And we do the same math. So we convert everything to percentages. We look at the organic ingredient totals. We have 95.9%. The question is, because we are at above 95%, do we qualify for organic certification? In this case, we don't, because the SF100 Green contains enzymes from BE microorganisms. And because we're not allowed to use genetic engineering or GMO techniques for organic certified products, this recipe would not qualify for 95% organic. The solution would simply be to change the improver from a non-organic certified improver to an organic certified improver. So, and then finally, we have there again a very similar recipe to the first one, but we've added a freshness enzyme into it this time around because we want to have a shelf life of, let's say, 10 days on this particular recipe. We do the same math where we convert everything to 100%. We look at our percentages, and in this case, we're at 96.3%. The Intense Fresh 540B is not organic certified. So the question is, do we qualify? And in this case, yes, because we have 96.3% of organic ingredients, but the non-organic ingredient is only produced using non-GMO enzymes. So even though it's not actually certified, the enzymes are on the list of approved substances, and they're not going to disqualify this particular recipe from being certified as organic. So in this case, we would be safe. Other than on the recipe side, there are some different regulations around how records must be maintained and how ingredients must be stored in the facilities, as well as you need an accredited certifying agent to certify your products. So all this information is detailed in the standard. You can go through that to get a better understanding of what would be required for you to certify a product as organic. One of the other challenges with organic is really around formulation. Often finding organic ingredients and having source of those is a huge challenge for manufacturers. Also, you're a little bit limited in what you can use because you can't use any of the GMO ingredients. So getting the right tolerance and volume is often a challenge, but there are solutions like the S500 Organic that can help give you the type of performance you would expect from a typical product. Shelf life can also be a challenge, but there are ingredients that Puratos sells that help with shelf life, like the Intense Fresh 540B and Intense Fresh 640O, both of which are freshness enzyme systems that will help you maintain the softness and the moistness in your bread. And then you would need to get some sort of a natural or organic mold inhibitor, like vinegar, to lower the pH and make your bread stay longer without molding. And then also making a tasty bread can be a bit of a challenge, but there are some solutions available from Puratos. We have soft grain amber grain organic, which is our grains that have been cooked in sourdough, which help maintain the softness of the bread because the grains are not absorbing the moisture from the dough and from the crumb rather. And they also give a great flavor because of the sourdough that's inside the grains. We also have several different types of sourdoughs that can be added in: organic sourdough Daphne, Sapori Daphne There's a liquid wheat-based sourdough, and it gives a San Fran sour type flavor profile to your breads. And we have a powdered organic sapori traviata, which is a rye-based sourdough that's been dried, and this typically gives fruity notes. So there are a lot of different options available from Puratos to help solve any types of organic needs when you're doing development of organic breads. That concludes the presentation. I'd like to thank everybody for, if you made it this far in the video, for your time, and feel free to reach out if you ever have any questions. Again, my name is Joe Layton, and thank you very much.